I think we're ready to get started. So today we're here at Nova Biomedical in Norwood, Massachusetts. We're going to be going over the Cellmetric XHT, which is the Solentim system designed primarily for whole well imaging. I'm going to walk through how to load our cassette stacker as well as a plate manually. And I'm also going to walk through the software and how we can operate within there. We have a few different camera angles here, so we'll switch between them. Hopefully nobody gets nauseous at those changes. A little bit about me. I'm Christine Mercadel. I'm the Senior Field Application Scientist here for the Solentim product line. And I've been working for Nova for just about 10 years. So a lot of experience with these systems. So we'll dive in from here. One thing you'll notice on this Cellmetric is we have this 10-plate temperature-controlled stacker, and this allows for semi-automated capabilities with your plate imaging. So in this case, we can load 10 96 or 384 well plates up into this cassette and load that in here. I'll walk you through exactly how to do that. So first, we open the door. You then want to make sure you position, let's make sure I get myself in camera. You want to make sure you position your A1 on your plates in this A1 position. And then you use the two handles on the cassette to load it in. Now this is a shell unit, so it's not exactly how it would look, but the process is essentially the same. If you wanted to run a plate manually, again, we're using a Corning CoStar where the cutout for A1 is present. You would open this door here and load your plate onto the plate stage. So from here, I think we can switch over to our software. And every time you open Studius, you'll have to log in. Now, you can use a standard lab login that everyone uses in your lab, or everyone can have their own individual logins depending on your preferences. Typically, if you're a more GMP-focused lab, you'll want each user to have their own login, but most research labs are using a standard lab one. So in our case, I'm just going to use our supervisor account. One thing to note here, you only have 3 attempts for password before you get locked out. So we always recommend having a backup supervisor account should you get locked out of your primary account. So once you input the password, Studius will open up, but we're not really seeing anything on here besides the navigation menu to the left. The main reason for that is we need to first either create or select a project to work with. So we can see many projects here that have already been created. This is a demo data set, so that's what those correspond to. But if you wanted to create a new project for your work, you would click that Add New Project button, give yourself a project name. It also automatically assumes today's date is the start date for your project, but you have the option to select a different date if you would like. You also have the option to input additional conditions, cell line descriptors, or any other descriptive material for that particular project. That information won't necessarily be particularly prevalent within Studius itself, but it will be present on any exports that you create, such as a clonality report or a conditional triage. Both of those I will go over later in our presentation. So in our case here today, I'm going to go into one of the projects we previously created. And now I'll walk through all of our navigation menu tabs. So at the top here, we can see seed, which is grayed out. This tab corresponds to our VIPS Pro instrument, which performs droplet seeding for clonality verification. So on that system, you can actually place a nanodroplet in a dry well, and it will take images of that droplet with a single cell. That can then be coupled with the whole well imaging on the Cellmetric X in the Scan tab. And this is where we'll actually be configuring our plates, selecting our scan types, and performing scans themselves. Our assay tab corresponds to the icon instrument, which is where we can perform different critical quality attributes, such as IgG titer assessment or glycan profiling. We then have our transfer tab, which allows you to track the scale up of your clones over time. So while it doesn't perform the physical liquid handling for you, it allows you to track within the software where those original wells from your 96 well plate went into perhaps a 24 well plate and then a 6 well plate up to bioreactors, so perhaps amber. Our view tab is where you spend the majority of your time. This is where you're actually looking at the data generated on the CMX. Our Analyze tab allows you to take all of that data and generate pick lists and some other functions depending on the instrument you're working with. Report is where we come for our clonality report, which compiles all of the images for your well of interest into one nice PDF report that can be used as part of an addendum for an IND submission. And then last, we have Manage, which is really for project and user management. But we're going to come back to our Scan tab here. So you'll notice to the left, we have all of these plates here configured. If you're just getting started for a project, you would need to click this Add Plate button down here. I'm going to walk us through that. So you'll notice in this submenu, you have the option to select Read Barcode, which removes the plate name field. One thing to note with the Cellmetric XHT, which is the one with the stacker that we have, it is actually required that the plates be barcoded. We made this adjustment primarily because we wanted to ensure there weren't any transcription errors when you're putting in 10 different plate names. You also have the option to input a manual plate name if you would like. And then next you need to select your plate type. Now it's very important with our system that you use a validated plate type because the software autofocuses for you. There are background dimensions built into the software when it's scanning the plates to give very high quality images. And because of that, if you attempt to run an unvalidated plate, you just may not get the quality of results that we would expect with a validated plate. So it's very important to select a validated plate. From there, you select your seeding date, which again, it defaults to today's date, but you have the option to input a different date. What that date tells the software is exactly when that plate started. So perhaps you seeded a plate 3 days ago, so you want to say, all right, day 0 for that project is Monday the 16th. So I'm just going to pick a plate at random to showcase how we walk through. Once you've got your plate configured or you select a plate that has already been scanned, you then want to come over to your scan types. At the very top, we have our Assisted Evidence of Clonality Precision Scan. This is a bright field only whole well image scan, primarily designed for 96 and 384 well plates. And this actually utilizes a neural network, so artificial intelligence, to help find areas of interest in the whole well. What we find this really helps with is reducing that analysis time when you are verifying clonality at that stage of your project. We also have a very recently developed assisted evidence of clonality scan that can also scan in green and red fluorescent channels. So with this scan type, you would get a bright field image as well as green and red channels if you wanted both. If you only wanted green, you could do that as well. This uses the same type of network where it's looking for single cells. It's just performing that function primarily based on the fluorescence profiling. Further down, we have 2 different types of confluence scan. Our precision scan, which takes around 12 minutes to run and gives you 3 planes of focus in the Z-stack, so top, middle, bottom. And then we have confluence throughput, which takes about 6 minutes to run and gives you 2 planes of focus. One thing to note, both assisted evidence of clonality scans will give you 3 planes of focus. We then have our manual evidence of clonality scans. These 2 scans are native to the instrument. So the other scans, which utilize neural networks for additional information, those are part of an advanced imaging package that is an upgrade for the system. So if you opted not to get those, you would just have these two manual evidence of clonality scans. The precision scan gives you 3 planes of focus, and the throughput scan gives you 2. Last but not least, we have our MSC confluence scan. This scan is really designed for confluence detection of more spindly cell types. So while we state MSC here, it doesn't only need to be used with mesenchymal stem cells. It can be used with other cell types that may just have different, more spindly morphology than a standard CHO, HEC, or iPSC cell line, which are the 3 validated types for these top 6 scans. So just to showcase what happens next, once you've configured your plate and you select your scan type, you'll then get this new submenu that shows well selection. Now the software always defaults to all wells, but you have the option to say, I only want to select the inner wells, I only want to select a few rows or a few columns. It's entirely up to you every time you scan on the system. So when you're ready, you've selected all your wells, you would just go ahead and click scan. Now we're not going to do that here today. I'll show you some data that we've prepared ahead of time, but that's really how easy it is to run on the Cellmetric X. You select your plate, select your scan type, select your wells, and click scan. I'll take a moment here to see if there are any questions that have popped up. All right. So we'll steam along ahead. So I'm now going to come down to our View tab, which is where you would want to navigate once you've finished going through your scans themselves. And I'm going to come down to this plate here. Now what we can see here is the most recent time point for this plate. So up here at the very top where I'm navigating my mouse, we have our history tree. And so this actually showcases every day within our project that some function was performed on this plate. So in this case, we're on day 14. And we can see here in green every well that has a colony that was detected by the neural network via the confluence scan. So I'm going to come back to our day 0 time point and our seeding time point. Now with the seeding map, we get this nice green and red. I apologize if any of you are colorblind, but we get this nice green and red map that shows green wells are single seeded, red wells either have multiple single cells or an aggregate, and white wells are where the system attempted up to 16 times potentially in that well and did not find a cell in any of them. So we'll come down here to this one. We're in well C5 right now. And what's nice with this is you can navigate into that droplet image, you can zoom in and out, and you also have the ability to go up and down in the focus planes. So you can see here very clearly where there's a single cell versus something like a micro bubble that doesn't change very much as we move through the focus stack. So we can then also move forward here through all of the time points for this plate. And I'll come here to our day 0 whole well image. Now the nice thing about this scan, so I have already annotated this data set, so we have this blue box here, but we can take this off to show you exactly what that looks like. Once you've run this AEC scan, you'll get a nice green box or red box, depending on what's present, on where there's an area of interest in that well. So in this case, because this was a fluorescent scan, I'm going to show you what the data looks like. So in this case, we just have our bright field image, but we can actually navigate through the different fluorescent channels. So this is our green fluorescent channel, and this is our red, of course, because the cell is not expressing in the red channel. We're not seeing anything there. But let's say I was at the end of my project and I was ready to annotate my data set to say, all right, these are the clones that I'm coming back. I want to verify that they are truly clonal on day 0 and create my clonality report. That is where I would come in. I would select this pencil, click cell, and then we can draw a little box around that cell. And you may notice, you may not have, but this annotation at the bottom here actually becomes illuminated. As soon as you add that cell annotation, it tells the software, I, as the end user, am marking this well as clonal, and I'm interested in having it in my clonality report. Now, what we can do then is move through all of our wells and all of our time points to look at each well and analyze them that way. Once we've analyzed all of our clonal wells, we can come down here to our report tab and click Clonality Report. And I have already created a report for us to work from. What this does is it actually creates a report for a page in that report for each time point for that well of interest. So in this case, we're looking at plate 2004, well C5. We have our droplet image with our single cell magnified area. We then also have our assisted evidence of clonality scan time point with the whole well, as well as that magnified area with the bright field image, green channel, and red channel. And you could do this, go through each image, so on and so forth, for every well of interest until you have gathered all of the data that you need. From there, you can export this report. It does export as a PDF. What I did today was actually export it ahead of time so I could show you all exactly what that looks like. So this is what our clonality report looks like for each time point. And you can see we just get continuing growth of that colony over time. It paints a really nice picture. This was a subsequent well that we have not gone over yet. All right. So once you've created your clonality report, you may say, all right, well, now I need a pick list to tell me which clones I want to move forward with to scale up to my 48 or 24 well plate. And that's where we're going to come to our Analyze tab. Now, our normalized titer and specific productivity analyses correspond primarily to our ICON instrument. So we're going to focus today on conditional triage. And what this actually allows you to do is create that pick list using some information that we've configured in here. So we can say, I only want wells that were tagged as VIP single-seeded, so the wells that were green. I only want wells that were tagged as clonal, so ones that I went through and verified were in fact a single cell on day 0. And I only want wells that have a confluence on day 14 of 10% or more. When you go to click this export button, that actually generates a CSV file with every well that meets all 3 of those criteria within the entire project. So it can be a really nice way to compile all of that data into one place. You can then hand that information over to your liquid handler for the actual clone picking itself. Once you have actually transferred those wells physically, you can then use the Transfer tab to track that scale up. So I'm going to click Manual Transfer here and Manual Definition. Now, the Import Triage file, when you get that pick list from our Analyze tab, you actually have the option to complete a destination plate and destination well location. In that case, you would be able to just import that file. But I find showing the manual definition is a bit more meaningful if you've never done it before. So I'm going to label our plate here, plate 1, 24 well plate, just under the assumption that we're scaling up to a 24 well plate. From there, you want to select the plate that you're scaling up into. So I'm going to select Corning CoStar 24. And then the transfer date, again, it always defaults to today's date, but you could select a different date if you wanted. Once I click OK, we'll have actually many more options here in terms of submenus. So any plate that has been configured in this project is an option as a source plate. Now, I'm going to pick one at random just because this is for demonstration purposes, but you have the option to manually select wells of interest from each plate. Another nice thing to note is that each individual source plate gets its own color. So once you actually fill those wells, they'll show up down here in the destination plate. Now, if we click Create, this will actually create a new plate within Studius. So we'll come back to View here. And we've got our new plate down here at the bottom, plate 1, 24 well plate. And we've got our nice 24 well plate here. But you also have the option to switch back to those original source plates and see exactly which wells have been transferred out and that entire original history tree. So it really gives you a nice visual representation of where those clones came from. And then you have the option as well, I did not mention this earlier, to scan those larger plate formats on the Cellmetric X itself if you wanted to quantify confluency at that point. So I'll take a moment to see if we have any questions that have popped up.
No questions at the moment, but please do ask everyone if there's something you want answered from what you're seeing today.
Yeah. I believe I've gone over most of the things I would like to today. I'll go over a few more things here while we're in the View tab. So at the top here, you can see this little caret next to plate name. You also have the option to order data differently. So you could do it based on time point. So this is going to be every single function that was performed in the project. Personally, I find plate name to be the most helpful because that's really how I think about organizing data in my mind. You also have the option to organize by plate type, but if you have many of one type, in this case, Corning CoStar 96, I find it a bit more challenging to really delineate versus by the plate name itself. All right. No questions. Okay. Well, I think based on that, we've gone over essentially all of the functions in the software, how we load a plate on the system, how we load our cassette stacker with our 10 plates, and yeah, I think we've gone over everything we need. So if there are no questions, I'll close early today and give everyone back some time.